Hello, my dear students. Welcome back to another learning lesson in my Physio-Pulse channels. Today we will discuss about the beta oxidation of fatty acids in a simple, easy to understand way. You will find the clear explanations, diagram, and important concepts here. Before we begin, if you find this video helpful, please like, share, subscribe to support the channel. Now let's understand about the beta oxidation of fatty acids. This process is known as the beta oxidation because the oxidation and splitting of two carbon units occurs in the beta carbon atoms, and the results of the sequential removal of two carbon fragments, and so it is called the beta oxidation. The oxidation of hydrocarbon chain occurs sequentially cleavage by two carbon atoms that was discovered by the Knoop in 1904. There are various stages and the tissues which are occur in the beta oxidations. There are specifically three stages. Number one, activation of fatty acids into the cytosol. Number two, transports of fatty acid into the mitochondria. And number three, beta oxidation proper in the mitochondrial matrix. So fatty acids are oxidized by most of the tissues of the body. Brain, erythrocytes, adrenal medulla cannot utilize fatty acids for the energy requirements. So how fatty acid is activated, that we will discuss now. Now first, fatty acid activation taking place in the cytoplasm. Fatty acids are converted into the acyl adenylate, and this acyl adenylate will convert to form the Acyl-CoA by the thiokinase or Acyl-CoA synthetase enzyme. The reaction occurs in two steps and requires the ATP, coenzyme A and also magnesium ions as required. The fatty acids react with the ATP to form the adenylate, which then combines with the coenzyme A and produces the Acyl-CoA. So two high energy phosphates are utilized here since ATP is converted to form the PPI or pyrophosphate. The enzyme inorganic phosphatase or pyrophosphatase hydrolyzes PPI into the phosphate molecule. So this is the reaction sequence: activation of fatty acid to the Acyl-CoA. How it is formed? This fatty acid, with the help of thiokinase enzyme and with the help of ATP, it will convert to form the acyl adenylate. Then acyl adenylate, with the help of CoASH, it will convert to form the Acyl-CoA. Now after forming the Acyl-CoA, this Acyl-CoA will transport into the mitochondria. How it can transport into the mitochondria, that we will discuss now. The inner mitochondrial membrane is impermeable to fatty acids. So a specialized carnitine carrier system, that is called the carnitine shuttle, which can operate to transport the activated fatty acids and from the cytosol to the mitochondria. Carnitine, what is carnitine? Carnitine is beta hydroxyl gamma trimethyl amino butyrate, which is synthesized by the lysine and methionine in liver and kidney. So it occurs in the four stages. Number one, first of all, acyl group of Acyl-CoA is transferred to carnitine, catalyzed by the carnitine acyl transferase 1, CAT1, which is present outer surface of the mitochondrial membrane. Now, acyl carnitine is transported across the membrane to the mitochondrial matrix by a specific carrier protein that is called the translocase enzyme. Now, this carnitine 2, that is CAT2, carnitine acyl transferase 2, it is found in the inner surface of the mitochondrial membrane. It can convert acyl carnitine into the Acyl-CoA, and ultimately the carnitine released returns to the cytosol for reuse by the translocase enzyme. So this is whole preparatory steps what we have seen, that the Acyl-CoA, with the help of CAT, it will combine with the carnitine to form the acyl carnitine. Then CoASH is and it can remove the CoASH. Now this acyl carnitine in the cytosol, it will transfer to the mitochondria with the carrier protein. Then acyl carnitine, with the help of again CoASH and CAT2, it will convert to form the carnitine and Acyl-CoA, and in the mitochondria this carnitine is reback into the cytosol again. So this is the whole preparatory steps of the beta oxidation of fatty acids. So I think you must have understand about the preparatory steps, definition, stages, how carnitine or how fatty acids or fatty Acyl-CoA transported from cytosol to the mitochondria or mitochondrial matrix. So students, we have completed our preparatory steps of the beta oxidations fatty acids. So if you have any query, any questions, any doubt, please give some comments in our comment box. And also we will learn in our next class, next lectures, specifically the steps of the beta oxidation as well as the energetics of beta oxidation, how many ATP can be produced from any fatty acids or fatty acyl-CoA molecules. So, if you find this video helpful, please like the video, subscribe to the channel, and turn on the notification bell so you do not miss any other biological lessons. Thank you for watching and see you in our next class. Thank you.